I'm very glad
Sorry,
to
yeah.
talk my
Mm-hmm.
okay, yeah,
Mm-hmm.
be able to talk our recent work to you and to the other audience. Okay, I'm Tetsuya Kobayashi from University of Tokyo. I'm belonging to the Institute of Industrial Science, but working on biology and also involved in other kind of institute in Tokyo University working on biology. Today I'm gonna talk about how we can use optimality theories to understand biological information processing. Okay, let me start. So first, I'd like to briefly introduce what we are doing so far. My lab is a laboratory for quantitative biology, and we are looking at mainly focusing on looking at the biological information processing. So my background is applied mathematics, but I think that you can safely recognize me as a theoretical biophysicist because I mainly use the mathematical techniques from physics and also sometimes from engineering. So we have been working on the different kind of biological phenomena listed here. We are looking at the information processing aspects of these phenomena so far. So because I'm theoretician, so we mainly construct mathematical tool or mathematical theories to analyze this kind of phenomena, and also we develop some informatic techniques to analyze quantitative data obtained in relation with these things. So in order to understand biological information processing, so in my lab, we are focusing mainly on the three factors. So one factor is the optimality of the information processing. So I'm very much interested in biological information processing because they looks very efficient, even compared with engineering system. This is very attractive aspect to me. But of course, in order to answer the question, whether a given biological system is actually effective or not, we have to compare its behavior with something optimal one. So we need a theory, optimization theory to predict or to derive what is the achievable optimal for a system under certain setting. So in the line of this kind of work, so today I'm gonna talk about how E. coli sensory system is designed optimally compared in terms of filtering noise. So I'm gonna talk about this topic later. But recently, we're extending this kind of approach to more complicated chemical sensing phenomena such as olfaction, and also the sensing of the antigens in immune system. So I don't have time to touch on it, but so we are working also on that. The second factor we are looking at is the kind of physical and also chemical constraint over biological system. As you know, all biological systems are composed of cells. A cell is the building block of biological system, and a cell is a collection of chemical reactions like this. So therefore, all biological systems should follow, of course, the physical and chemical and also some dynamics law. And therefore, we have to understand more what can be done, what can be implemented by using chemical reaction networks. And also we have to understand associated thermodynamics in order to account for the dissipation that should be paid for realizing certain kind of function by using chemical reaction network. So in this line of work, we recently developed a unified theory of chemical reaction network and thermodynamics that can be applied for nonlinear and also non-equilibrium phenomena. And as a application of this theory, so we also derived the last year a thermodynamic condition under which self-replication can be realized. So this is purely thermodynamic theory when we can when the self-replication becomes possible. Okay, the last factor we are looking at is the evolution and also the ability to self-replicate of the cells or biological systems. Actually, compared with engineering system, we have also engineering information processing system. So big difference between the biological system and engineering system is that biological ones have been shaped by evolution.
Mm.
And also biological system has the unique ability to self-replicate. So these two factors may affect the possible behaviors of biological system, and I want to understand it. Especially we are trying to formulate or and understand evolution as a kind of information processing by using large deviation theory and information theory. And so the motivation is to understand the two aspects of the adaptation in biological system. One is the individual adaptation that is done by ourselves. So our brain is designed to predict future and select adaptive behavior as a single agent. But on the other hand, in evolution, by natural selection, the adapted organisms are selected kind of posteriorly. And so these two are quite different, but so coexist in many biological phenomena. And so we want to understand this kind of things. But anyway, so these are the brief introduction of what we have been working on. And so today's work in today's talk, so I mainly focus on the first one. Okay, and so I prepared two topics today, and one is the optimality of bacterial chemotaxis, E. coli chemotaxis, that we did in these two papers recently. And the second topic is motivated by the first one. So second one is more theoretical and purely theoretical. We developed a new theory to address the kind of bounded rationality of biological agents in terms of information processing and controls. Okay, let me start with this first one. So in this first part, so I gonna look at the chemotaxis by E. coli. And so this is a movie of swimming E. coli cells. And from this movie, we can see that, so we have two modes of swimming. The one is a kind of ballistic swimming here, for example, that is called run. And also sometimes that ballistic swimming is interrupted with the random reorientation that is called tumbling. So there are two modes. So if these cells are exposed to a attractant gradient, a cell try to modulate the frequency of this random tumbling. So if a cell feels that it's going down the gradient, this cell increases the frequency of random tumbling. If it is lucky, then it will be reoriented towards up the gradient. Then, so if this cell feel that it is going up the gradient, then this cell suppress the frequency of the random tumbling. so enable it to go up the gradient. But of course, there is a stochasticity in cellular behavior. Then sometimes, so this guy does tumbling again. But on average, by suppressing the tumbling when it is going up the gradient and increasing it when it is down the gradient, then so this cell can swim up the attractant gradient. So the reason why we are looking at this E. coli chemotaxis is that, so this is the sensory system characterized best quantitatively, I think. And so we already know major molecular network and molecular modification or something like that involved in this E. coli chemotaxis. Depicted here. And also it is not so difficult to monitor the motor movement, the rotation of the motor that can determine the ballistic run and also the random tumbling. So by attaching a cell to a glass, then we can monitor the motor movement by a microscope, by using a microscope very easily. Therefore, we can measure the quantitative response of this cell to the change in the ligand. So here we have step like the input to the system, and then so if suddenly the ligand concentration increases like this, then this cell increase the frequency of the ballistic run like this, and then get adapted to the original position. So by using bioimaging, we are also able to monitor the response of signaling molecule involved in this network. So because of these quantitative data, so for E. coli chemotaxis, we have a standard biochemical model proposed by Semis and Berg in 2008. So this model is a simplification of more complicated statistical mechanical model that is called MWC model. But so this model is good enough to reproduce and also explain various kind of experimental data. So this model were used in these papers. So this model has the attractant concentration and also the receptor activity here. So A is a receptor activity defined like this, and it is a function of the chemical free energy difference of the receptor state, activated state and inactivated state. And this F is a function of the ligand concentration, attractant concentration, and also the methylation level of the receptors. And so this M should be changed depending on the receptor activity. And unfortunately, from this model, so the functional form of this F cannot be derived mathematically, but so they used experimental data here to fit this function to experimentally estimate this function F. So because this F is decreasing, so this F introduce a negative feedback in this system. So we have very nice model already. But we still have a couple of problems, unsolved problems in E. coli chemotaxis. Among others, we focus on the effect of stochasticity and noise. As you know, we have intrinsic stochasticity in cellular system, for example, gene expression. And also we have the stochasticity in the binding of ligand and receptor. So this is not from E. coli experiment, but so this is an illustration how the ligand binding to receptor is, how stochastic it is. So this is a amount of the receptor bound to the number of the ligand bound to the receptor as a time series. It's quite noisy. So as I said, E. coli have to judge whether it is going up the gradient or down the gradient in order to modulate its swimming behavior. It is not difficult if the E. coli sense ligand concentration deterministically. So because so this is a kind of computation of the time derivative of the ligand concentration around the swimming. But okay, any question?
Oh no, it's great. Yeah, thank you.
So, but so E. coli have to use stochastic receptor interaction with the ligand. Therefore, actual signal that a cell can obtain should be very noisy. So because the derivative of noisy signal will amplify the noise rather than the signal, so it is not trivial to judge whether it is going up the gradient or down the gradient. So my question is whether E. coli chemotactic system is designed optimally to cope with such kind of noise. So to address this problem, so our approach is a bit different from usual one. So usually, especially biophysicists, start with modeling actual biochemical system and investigate its behavior theoretically. But by this kind of modeling, we cannot answer whether this system is close to optimal or not. Of course, we can understand how this guy behaves, but so therefore we start with the defining the task that the system is solving, may be solving. So that is, in this case, that is the inference or estimation whether it is going up the gradient or down the gradient for the E. coli cell. Then by using the optimization theory, we derive optimal system to solve this task. Then finally, we compare this optimal equation or optimal system with the actual biochemical model, whether they are close enough or not. So this is our approach to investigate the optimality of biological system. So to this end, so we first define what kind of task a E. coli is solving. So we suppose that E. coli want to know whether it is going up the gradient or down the gradient. So xt is a variable representing it. So if it is going up the gradient, this xt is +1. If it is going down, then it is -1. So we define the axis around the gradient, xi. Then the position of the cell at time t is obtained by this differential equation. This v is the speed of the swimming. And so here we have x. Then, so we model the switching of the direction, swimming direction, by using two-state Markov model here. So R plus and R minus, the frequency between the switching from going down to going up and going up to the going down. So therefore, on the time, so this cell swim up the gradient, down the gradient, up the gradient, down the gradient due to the flipping of swimming direction. So cell want to know whether it is swimming up or down the gradient. And on the axis, there must be a gradient of attractants. So we suppose that there is a exponential gradient around this axis. So then finally, this y is the stochastic variable representing the output of the receptor. So this y is a function of attractant and also the receptor noise. So its time derivative is very noisy. Here we have it's a Wiener process. And so we have log here because we want to compare our result with the biochemical model later. And so this log appear already in this biochemical model. So therefore, So this log come from the biochemical model already proposed. So given this information, what we have to do is to derive optimal dynamics that infer the swimming direction x only from these noisy signals. So we can use the theory of continuous time nonlinear filtering. Specifically, we use the Kushner equation that is equivalent to Bayesian filter to obtain the dynamics of Z. So this Z is the posterior probability that a cell is going down the gradient estimated given the whole history of the noisy sensory signal. So this Z should be computed by this stochastic differential equation driven by this noisy Y alone. So here. So this is a nonlinear equation, but even though this equation is driven by this noisy one, so Z behave like this. So on in the gray region, so a cell is swinging up the gradient. So in this region, so Z goes down to 0, meaning that so this Z is estimating that it is going it is not going down the gradient, because that is a probability that it is going down the gradient. And so in the white region, so that becomes close to 0, and then again something like this. So this means that so that can recover the information of the X only from this noisy signal Y. So this is the optimal way to filter the sensory noise. So now we have the optimal equation here. So let's compare this equation with the standard biophysical model proposed already. So they look different at the grace. But so let me introduce a variable transformation. So from the posterior probability Z, we convert it to the posterior log likelihood ratio theta. Theta is defined like this by using Z. And by solving this equation inversely, then so we can have the representation of Z with respect to the theta. And the dynamics of theta should behave like this. So this is very simple rule of the derivative. And then so by plugging so this equation here, and also by formally integrating this equation, we can obtain an equation for theta like this. So here we have ligand concentration. This is embedded in here. And also we have noise embedded in here. And also we introduce the mu additionally. So this mu should follow this equation. The right-hand side of this mu basically come from this term and also this term. So this one come here, and so this term come here. Okay, then after this variable transformation, it is should be easy to see that the optimal equation correspond to the biophysical model. So the posterior probability can be associated to the receptor activity A, and log likelihood can be associated to the free energy difference F. So here we have ligand concentration, and so mu can be associated with the maturation level. So the good point in our optimization theory is that, so right-hand side here is explicitly obtained only from the theory. So in the case of biophysical model, this F was not derived theoretically, therefore they use experimental data to infer the functional form of this F. So therefore, so in order to further test the validity of this correspondence, we can check whether so this F from experiment can be close to this functional form. So we did it. So black points from experimental data, and red line is our function on the right-hand side. So our function only has two fitting parameters. One is a scaling of y-axis, and the other is, this is a kind of bias of the swimming direction towards up or down. So this parameter just shift the this curve along the x-axis. So even with only two fitting parameter, so our function can nicely be fitted to experimental data.
Nice.
So this result suggests that E. coli sensing system may be designed to filter noise optimally. So however, so we are ignoring one important fact, that the E. coli cell can control the swimming behavior of itself based on the sensory signal. Then actually in our modeling, we suppose that we model the swimming, the switching of swimming direction by a two-state Markov model. So this is independent. So therefore, this switching was assumed to be independent of the sensory sensing process of the cell. But in reality, after sensing, the cell modulate its motor control. So this leads to the reorientation of the swimming. And so therefore, the sensed information should affect the swimming direction. Therefore, there is a kind of feedback loop in the system due to the sensing and also the subsequent control. So in order to accommodate treat this kind of fact, we have to use the theory of partially observable Markov decision process. So specifically, we have to additionally model this process of the modulation of swimming direction. So here we use a Poisson point process to represent the tumbling event sequence. So this spikes the tumbling event, and we suppose that cell can modulate the frequency of the occurrence of these events. So this R is the frequency. And so our assumption is that this frequency should be the function of the whole trajectory of sensory signal. So this is very general setup theoretically. So cell can use any kind of information obtained by sensory system in the past to modulate its tumbling frequency. So in order to apply the control theory, we have to define the objective function of this system for the cell. So the objective function has two components. One is the utility function. So here we use average speed of the cell around the gradient, up the gradient. So this is average speed. So this is average. And this is a cost function. I will discuss about it later. So given this objective function, so the goal, just a moment, yes, the goal is to maximize this objective utility function with respect to this tumbling frequency function. So this is, so we get the optimal control strategy. So, but to solve this, I'm sorry, just a moment. In order to solve this optimization problem, we have to specify the cost function. So actually, this is a problem. There is a problem in defining the cost function for control. Then actually, so the problem is that so we do not know the relevant cost for biological control. in usual control theory, people often use quadratic cost, but that sounds not so good for biological system. And more practically or more technically, so we have to obtain the optimal tumbling rate R. So this should be non-negative. And if we use the quadratic cost, then so negative R may cancel out in this quadratic cost. Therefore, by solving naively the optimization problem, we may end up with the negative R or something like that. This should be avoided. Mm-hmm. So we have two problems. So to address that, so we adopted Carbac-Ribera cost proposed by Todorov in 2009. The idea of Carbac-Ribera cost is that we define the cost of the control by the Carbac-Ribera divergence between path probability of the reference state and also controlled state. In our case, the reference state corresponds to the stochastic tumbling dynamics without any control. For example, the constant rate tumbling rate situation. And then by the control, this tumbling rate is modulated. So therefore, we have the biased probability by the control. So we evaluate the cost of the control by the difference between these two. And so by employing the Carbac-Ribera cost, so we can have very nice representation of the instantaneous cost for control as a function of the R here at time t. Mm-hmm. And also this, so this functional form automatically guarantee that the R is always non-negative. So then I skipped this detail, but so basically, so by using technique of the partially observable malcondition process, we solve this problem by introducing HJB equation for the optimal control and value function or something like that. So by considering the control downstream of the sensing, so our previous result for the right-hand side of the maturation level will be is updated like this. So here we have at the scaling factor, the position of the scaling factor. So we have the optimal control rate of the swimming of the tumbling here. And also this zeta bar, this was a parameter in the previous case, but this was replaced with 1/2. So meaning that swimming direction is the even, swimming up and down occur evenly without control. So this 1/2 means that's kind of prior information, but that kind of swimming direction is biased by the control here. So this R cannot be derived analytically, but we can obtain it numerically. And so this R should look like this as a function of Z. So this is a probability, posterior probability that it is going down the gradient. So therefore, if a cell is very sure that it's going down the gradient, then so this R increases. So this functional form tells us that kind of thing. And if the cell is very sure that it is going up the gradient, then so tumbling rate should be suppressed. So this is very consistent and also very intuitively reasonable to control the tumbling frequency to swim up the gradient. So by using this functional form here, then we can also fit the experimental data nicely by using this function. So in conclusion, so our analysis suggests that E. coli sensing system may be designed optimally to control itself by filtering noise in the sensory step. Okay, so this is the first part of first topic. And the second topic was motivated by the result of this one. So actually, so E. coli chemotactic system is one of the simplest sensory system. So we want to extend our approach to more complicated biological information processing. But to address that, so we realized that conventional optimization theory is not appropriate. So this is the reason why we considered a new theory for this problem. So what is the limitation of the conventional optimization theory? We think that is the ignorance of resources required for realizing optimal behavior. So we have many optimization theory in engineering, for example, information theory, estimation theory, and also control theories. In control theory, for example, so we very faithfully model the target or environment that we want to control. So we consider stochasticity in the environment or target, and also uncertainty in this system. And we also consider control cost when we control this target system, and also we takes into account the noise when we observe this system. But we totally ignore the cost at the agent state, or in other words, on the side of the controller. So in the control theory, we want to design controller to control the target system. So, but by using the conventional theory, what we can obtain is that the controller, so the conventional theory requires the controller or agent should have infinite memory, and also this agent should behave without any noise, without any cost. So these things were not considered in conventional one. So in the case of the filtering problem, for example, so in the first part of my talk, we say that E. coli can, so we introduce Z. That was the posterior probability going down the gradient. So in that case, so a cell was estimating it is going up or down, therefore it's a binary information. That is very simple. But if a cell wants to estimate the direction, for example, theta maybe would be easy to understand, theta. So if a cell wants to estimate the direction of the food or the gradient, then the cell needs to store the posterior probability for this direction. So this is a function, therefore, ideally, the agent or a cell should have infinite memory to memorize it. And also, in order to compute the posterior probability, a cell or an agent should compute right-hand side. This is Bayes' rule. So to compute this right-hand side, noise should not be introduced. So therefore, the agent should be able to behave without any noise. And also, as you know, any cost for computation of the right-hand side was considered in this case. But we know that all biological systems are finite and also bounded in terms of available resources. Depending on the amount of the resources that can use, the behavior, information processing of these organisms may change. Of course, if the more resources are available, then more complicated and efficient information processing seems to be realized. like this by using, well, in the case of our brain. So the conventional theory consider only the ideal case that so available resource is almost infinite. So this kind of resource limitation is very interesting from evolutionary biological context, because depending on the available resources, the information processing system have evolved. So we may ask whether this evolution is continuous or punctuated. So this is very attractive, I think, the problem. So in order to address this issue and this problem, so one of my students constructed a new theory for estimation and control under limited resources. The key idea is very simple. So he explicitly modeled the agent here. So usually in conventional control theory, we only model the environment by using stochastic differential equation, and also we model the noisy sensing and also the cost of the control. But he also introduced a model for the agent, so by using stochastic differential equation, and also he considered the optimization of this agent by itself by introducing another control parameter B from the agent to itself. So we explicitly define the behavior of the agent by a equation that we can consider finite memory. For example, if we assume one-dimensional equation, then so a cell can use one-dimensional memory or something like that. And here, because we have the noise term here, so the effect of noise is accounted here. And finally, so we solve the joint optimization problem. So in this case, the optimization objective function should be a kind of cost. Therefore, here we have the minimization. We want to optimize the control from agent to the environment. So this is a usual one, but also we optimize the control from agent to agent itself. So this additional control optimize the agent itself, how to memorize the sensory signal and how to behave in this with respect to that. So this is a formulation so we introduced. So, but this is not difficult. So this is very intuitive. So problem is how to solve this this problem with resource limitation. So this is a resource limited partially observable stochastic control. So the idea is to solve this problem is first. So we introduce an extended state. Extended state S is a concatenation of the state of the environment and the state of the agent. And also similarly, so we defined extended control for the environment and also the control for the agent itself. By introducing these extended state and control, so we can combine the two equations we can represent then by single equation formally. And so here we have extended control. But the point is that, so this extended control is function only of the Z. So that Z is the state of the agent. So agent cannot directly know the environment. So it can use its internal state to determine the control. So if this Z is replaced with the whole extended state, then so this problem is reduced to the so-called completely observable stochastic control problem. And so that problem can be solved by using dynamic programming on state space. In this case, we have to solve HJB equation in the form of partial differential equation. This is accessible. But by considering this limitation here, and the problem becomes very difficult formally. So because in order to solve this problem, we have to solve dynamic programming on probability space, because the state of the environment should be inferred somehow by only from the state of the agent. Therefore, HJB equation now becomes functional differential equation. This is almost impossible, even numerically. But the trick my student introduced is to switch from dynamic programming principle to the Pontryagin minimum principle. So this trick was recently utilized in the mean field control, for example, in this by this guy. So by switching from switching to the Pontryagin minimum principle, then the effective HJB equation becomes the partial differential equation. So this is accessible. So this is the trick. So more roughly speaking, so this is the Bellman dynamic programming principle. And the state space, we have the value function omega here, and then so we can convert this to the Pontryagin principle. Then in this case, so partial differential equation can be converted to the ordinary differential equation. In our case, we have to solve the similar problem for on the probability space now. So the Hamilton HJB equation from Bellman principle is the functional differential equation, but by introducing the variation of this functional value function, and we define the variation like this, then so we can convert this, we can obtain the Pontryagin principle like this. So the point is that here we have the equation for omega. This is very similar to that. And also, if this H is a function, is kind of averaged with respect to the probability, then this variation produce the H, the term before averaging with respect to this P. And therefore, then we can recover something very similar to this equation eventually. So this is a very intuitive idea behind our approach. So I skip this one. So finally, so we tested our algorithm by using so-called LQG problem, linear quadratic Gaussian problem, as well as non-LQG problem. So for LQG problem, so we compared our algorithm, so in which the memory of the agent is optimally controlled. So blue one is the control without memory optimization, meaning that optimal control was derived as a function of instantaneous observation. So in this case, the controlled system, controlled state is very unstable. Blue one is unstable, but by considering the memory optimization of the memory of the agent, then so the control is stabilized. And also we should mention that from our formulation, we can rederive the Kalman filter equation and also associated linear control result. So meaning that so our formulation is very much consistent in that kind of thing. So in this problem, so the agent should, starting from this point, should go to this goal, but there are two obstacles here, and in this region, so the agent has a penalty in objective function. So this is non-quadratic, non-LQG problem. So therefore, optimal control should avoid. So optimal control should be avoiding so this obstacle, and actually by our algorithm, so we can obtain such kind of trajectory after control. But if we use local LQZ approximation, so if we approximate non-LQZ problem locally by using LQZ formulation, then so the result is something like this, the blue one. So this kind of algorithm cannot avoid this obstacle. And also the objective function, the value of objective function is much higher in this approximation. Okay, so this is a demonstration of our theory. And actually, so at this point, up to this point, so we constructed a general theory. And so my student is now working on applying our theory to biological problem. For example, tracking of the target by agent by sensing, for example. So this is an example. So this white blood cell is tracking and bacterium by sensing itself, sensing this guy. So this kind of problem can be formulated like this. So the agent sense the target and then control its motor to minimize the distance between target and agent. And also this agent should optimize the dynamics of its memory update. So this kind of problem maybe can be addressed by our problem. And also we are interested very much in the evolution of the information processing. We want to investigate that kind of thing by using our theory. For example, what happens if we increase the available resources? Is there any kind of transition in the information processing architecture or something like that? So we want to address this kind of problem in our future work. Mm-hmm. Okay, I think it's time to close. And so finally, I'd like to thank my student, my collaborator. The first topic was done by Kento Nakamura. He's now working for Riken. And also the second part was done by Takehiro Totori. He's now writing his thesis. So thank you for your kind attention. I'm happy to take any question or comment.
Okay, thanks so much. So, okay, let me do this. Let me stop